Hello everyone and welcome. In this video we're going to be checking out 9700 variant 1 of October-November 2022, the MCQ. There are timestamps available in this video if you want to skip to a specific question you would like to check. And if you have any question in biology, feel free to leave those down in the comments. And also if you'd like to share with us resources for the other students to check and maybe use, this way we can both help together as many biology AS students as possible. Let's get started. So for question number 1, the photomicrograph shows part of a plant cell. What's the magnification of the cell? Now I will not be able to do this on the iPad over here, but what I can do is just explain to you the steps. First off, the magnification, in order to calculate it, you have to be familiar with the triangle of image length, actual length, and magnification. So MIA. Magnification equals to image length divided by actual length. If you want to calculate actual length, you divide image length by magnification. If you want to calculate image length, you multiply magnification by actual length. So multiplication on the two sides that are next to each other, however division when they are on top of each other. Now, of course, don't forget that you have to measure the cell, like the scale bar length, so you can calculate the magnification using the scale bar length and then convert the scale bar length from millimeters to micrometers. So let's assume, for example, that the scale bar is of length of 20 millimeters, right? 20, in order to convert it to micrometers, you have to multiply by 1000. If you want to go back from micrometer to millimeter, you will divide by 1000. And so here, the image length is 20,000. If you divide that by 5, you should be able to get the magnification, and this is the sign for the magnification. And so here, using the answer that you will get by measuring the 5 micrometers on paper, the scale bar on paper, you'll be able to calculate the answer for number 1. Now, which row matches each organelle with its function? Microtubules make cilia and spindle. Microtubules do make cilia and spindle, actually, they are what forms it. Radicular reticulum separates some process from the cytoplasm, that is true. Golgi bodies make lysosomes, that is correct. And the centrioles assemble the spindle during its nuclear division, that is their function. It's assembly or organization of the spindle fibers during nuclear division. Let's look at the other answers and see if they have anything wrong. Move vesicles within the cell, that is true because they make the cytoskeleton of the cell. Assembles amino acids to make proteins, that is like the role of a ribosome. Contains enzymes for destroying worn out parts of the cell, okay, sure, during autophagy. Moves chromosomes apart during anaphase, no, that's the function of the microtubules or the spindle fiber. Okay, move chromatids apart during anaphase, okay, that is correct. And modifies proteins which may be released from the cell, true, but it's mainly a function of Golgi body. Makes glycoproteins, correct. Move cilia, centrioles do not move cilia. The cilia are going to move because of the movement of the microtubules, or let's say the microtubule doublets alongside each other, and the fact that they have these dynein protein arms, but not the cilia specifically. Yes, the centrioles form this basal body, which is going to be the structure at the bottom of the cilia, and it is what forms the microtubules in the cilia, but it does not move them, and that's why this is wrong. Okay, here move individual cells, that is also wrong because when they do not move on individual cells, their only function is actually to divide as part of centrosomes during mitosis, and also basically to organize the microtubules of the spindle during nuclear division. So it has nothing to do with moving individual cells, and also to form the basal body at the bottom of the cilia. And so my answer to this will be A. Number 3. Where are ATS ribosomes found? ATS ribosomes are found, they're not found in chloroplast, they're found in cytoplasm of eukaryotes, not in mitochondria, and not in cytoplasm of prokaryotes. Hence, the answer is 2 only. Which feature present in an organism enables it to be identified as a prokaryote? So, we can say cytoplasmic DNA, sure. Cell wall, no, because plant cells have cell wall. Protocysts, their cells have cell walls. Nucleus, no, prokaryotes do not have nucleus. In fact, the name means before nucleus. Karyote means nucleus, and it's before nucleus. Ribosomes, they do have ribosomes, that does not, however, distinguish them and allow us to identify them as prokaryotes, because eukaryotic cells, as we've seen here, actually have ATS and 7TS ribosomes as well. What's the function of the nucleolus? The formation and breakdown of the nuclear envelope. Actually, no, the nucleolus is responsible for formation of ribosomal RNA, which forms a part of the ribosomes. And so the synthesis of ribosomal RNA, that is correct. However, it's not the synthesis of ribosomal proteins. Never pick this option. I know that we usually say that nucleoli, they form ribosomes, but they do not exactly form the ribosomal proteins. They, I think, maybe already found in the nucleus. So let's say D. Okay. 6. The diagram shows the results of a number of tests on solution of biochemicals. The solution of biochemicals, which are basically written here. Benedict's solution is blue. Non-reducing sugar test is positive. Interesting. Iodine test is also positive, and biuret test is blue, so it's negative. And this is also negative. So, which substances are present in a solution? We have non-reducing sugars and starch. So that's not this answer. Protein, non-reducing sugar, and starch. Well, no, we do not have protein, however, non-reducing sugar and starch, which is A. Interesting. Okay. 7. Which diagram shows the ring of beta-glucose? For beta-glucose, you're going to notice that the arrangement of the OH groups on carbon number 1 to 4 is going to be up, then down, then up, and then down, which is up, down, so that's A. And however, up, down, this is alpha glucose. And for this one, down, I'm not familiar with this sugar. Down, up, down, again, another sugar that I'm not familiar with. Which row correctly matches the example with type of molecule? The type of molecule, disaccharide, we have fructose. No, it's not a disaccharide, it's a monosaccharide. Starch is a polysaccharide. Maltose and sucrose, however, are disaccharides. Macromolecules, ribonucleic acids and cellulose are macromolecules. Monomers, sucrose or fructose, obviously fructose, never sucrose. A polymer, cellulose or collagen, well, obviously this is not going to be the right answer, so the answer is D. Number 9. The enzyme alpha amylase hydrolyzes amylopectin, but it's not able to hydrolyze some of its glycosidic bonds. It only hydrolyzes 1,4 glycosidic bonds, interesting, and it's not able to hydrolyze the last bond of a chain. Okay, it says not able to hydrolyze the chains in 3 units attached by 1,6 glycosidic bonds to another chain. So which glycosidic bond can be hydrolyzed by alpha amylase? Interesting. So we know that basically anything within this 3 carbon unit 3 unit chain attached by 1,6 glycosidic bond can be broken down, hence A can be cancelled, and so will B because it only hydrolyzes 1,4 glycosidic bonds. It can hydrolyze C because it's a 1,4 glycosidic bond, however it cannot hydrolyze D because it is the last bond in the chain, right? So that's why the answer here is C. Molecule X is a lipid, and which row is correct for the molecule X of a triglyceride? Molecule X contains 1 unsaturated fatty acid. Well, we can see that actually it is saturated, so the answer is wrong. No ester bonds, well, that is correct. Actually, no, we can see that we do have an ester bond right here, right? And so there is actually 1 ester bond, 1 fatty acid. Triglycerides have 3 fatty acids, that is correct. Number 10. For question number 11, bread contains a mixture of polypeptides known as gluten. 2 of the polypeptides found in gluten are glutenine and gliadine. So what statement describes the tertiary structure of the protein? Disulfide bonds form between glutenine and gliadine. Well, that actually describes the quaternary structure, so we can eliminate that. A large proportion of amino acids in gliadine are glutamine. Well, this describes the primary structure more than it does describe the tertiary structure. Alpha helical sections are found in glutenine and gliadine. This describes secondary structure. Amino acids with hydrophobic R groups are found on the inside of glutenine. Exactly. This is known as the bond. These are hydrophobic interaction bonds, and basically they are part of what maintains the tertiary structure, so they're very important. 12. A student was provided with 2 test tubes, one containing 10 centimeters cubed of solution P and one containing 10 centimeters cubed of solution Q. When these solutions were mixed together and left for 24 hours, the concentration of P decreased. However, the concentration of Q remains the same, so we can assume that P is being broken down by Q, right? And so the student wrote the following conclusions. What conclusions can be supported by the information? That P may be the substrate for Q, that is correct. Q is a biological catalyst, that is correct. And P may break down over time, well, that is true. So we can say 1, 2, and 3, which is A. For number 13, what statements about correct for lock and key hypothesis and the induced fit hypothesis of an enzyme action? So for both this and that, we can see the substrate is the same shape as the active site. Actually, it's wrong for both of them because the substrate is complementary in shape, or the substrate's binding site, to be specific, is complementary in shape to the enzyme active site. However, it's not the same shape because then it cannot enter the active site. The substrate is held in place by the active site by temporary bonds. Correct is temporary hydrogen bonds. And the enzyme and substrate sometimes change shape slightly as the substrate enter molecules. That is not wrong. So basically, it's only 2, which is C. Number 14, a student investigated the rate of an enzyme activity with increasing substrate concentration. The experiment was repeated with the addition of a reversible non-competitive inhibitor. a graph was plotted to show the results. Which graph represents the results of the two experiments? So obviously the rate of enzyme activity, we can see that for a non-competitive inhibitor, this is the shape of the usual graph. And if we change it to a reversible non-competitive inhibitor, the shape really doesn't change, because what they mean by a reversible non-competitive inhibitor is that instead of just being stuck onto the enzyme forever, it actually can leave. And so the enzyme, it can leave the enzyme afterwards, right? But what happens is the effect is still the same. That it leads to the denaturation of the enzyme, it leads to the breakdown of the hydrophobic interactions, and the hydrogen bonds that are maintaining the tertiary structure, they'll all be broken down. Right, the same effect, however, it just is able to leave. And so the effect is going to be the same. It's going to reduce the Vmax because the substrate, however you increase the concentration, will still not be able to bind to the enzyme anymore, because let's imagine the shape of the substrate being like this. The enzyme active site has completely changed, despite whether or not it's a reversible non-competitive inhibitor. It's the same effect, and so the Vmax will be lowered, and hence the answer is B. Cuboidal epithelium cell has a length of 2 micrometers. The cell is an approximate cube shape. What's the most accurate estimate of the surface area to volume ratio of this cuboidal epithelium cell? So the most accurate estimate of the surface area to volume ratio, we can say basically, because it is a cuboid and we need to calculate. So first off, we need to know that the surface area of a cuboid is 6 times side length squared, right? And then the volume of the cuboid is side cubed. And so this is 6 multiplied by 2 to the power of 2, and this is only 2 to the power of 3. Right? And so here, this is going to be 24. Here it will be 8. And 24 divided by 8, which is how we get the ratio, will be 3:1. And that's why the answer is D. Now 16. Which statement is correct for facilitated diffusion and active transport? Both of them. Let's see. As the direction of the concentration gradient changes, so does the direction of movement of molecules. That is false, because active transport is always going to be moving against the concentration gradient. And I suppose if you flip the concentration gradient, that doesn't really matter, because it's just going to keep doing the pushing against the concentration gradient. Okay, molecules always move at the same rate as simple diffusion, not necessarily. Okay, specific molecules are transported across a membrane. This is correct, because the protein carriers are specific. That's basically this. They're specific. They can only carry a specific type of substance. The molecule ATP is required to move specific molecules quickly through proteins in the membrane. That's wrong, because the facilitated diffusion is a passive process. It does not require ATP. Hence, the answer is C. Number 17. Equal-sized potato pieces were placed into a test tube and covered with a sucrose solution. The test tube was left for 30 minutes and all other variables were standardized. After 30 minutes, the potato piece had not changed in size. What can be concluded from this result? So we can see basically that the water potential in the potato is the same in the potato and in the sucrose solution, and there is no movement of water. No, there actually is no net movement of water. That is exactly what I'm looking for, because you cannot really go by explaining the concentration of sucrose is the same in the potato, because the potato also has other stuff inside of it, not only sucrose. It has maybe mineral ions and other stuff that make it concentrated, and so it's not just the concentration of sucrose that dictates the movement of water, right? And so 18. How many of these processes could produce genetically identical cells? So genetically identical cells are produced in the growth of plant roots. That is correct. Okay. And the replacement of dead human skin cells. That is correct. Okay. Repair of damaged muscle tissue. True. Asexual production of corals. Correct. So the answer is D. Now, which row shows the correct number of each component of a single chromosome at the end of prophase of mitosis? So, of a single chromosome in prophase, we're going to have only 1 centromere, 2 chromatids, 4 polynucleotide strands, and 4 telomeres, because the chromosome is going to look something like this. This is a centromere, this is a telomere, and here we have a chromatid, and in each chromatid is made of 2 polynucleotide strands. Which processes occur during interphase? So during interphase, we have DNA replication. That is correct. During S phase of interphase, microtubule organization. Well, actually, microtubules are not produced all the way until prophase, and so I would say the answer, so 2 is wrong. Synthesis of ribosomes. Well, ribosomes, yes, they are produced, they're organelles that are produced, and they could be synthesized during G1, first course period. So it's 1 and 3 only. Now for 21. Embryonic stem cells are able to replicate continuously. What happens to the telomeres during the repeated mitotic cell cycles of embryonic stem cells? Well, basically, telomeres for stem cells have the enzyme known as telomerase. Telomerase is an enzyme that is going to avoid the breakdown of telomeres and avoid basically them shortening in length in order to basically allow the cell to undergo mitosis continuously. Because once you lose telomeres are like these protective caps of chromosomes. Once you lose them, then you're gonna eventually start to lose the genes. Like basically, during every single cell division, DNA polymerase does not really continue to replicate the strand until the full end of the strand. That means basically some part of the strand is going to be lost, right? And that's why telomeres are present in order to prevent it from being lost at the gene site. Because if you lose part of the genes, then this is when the cells are going to die. It's going to be known as programmed cell death, apoptosis. In order to avoid this happening in stem cells that divide continuously, because you need lots of cells to replace all the damaged cells, and how often skin gets damaged, it needs to be replaced continuously, and so that's why their length remains the same, because of the enzyme telomerase that increases the length of telomeres and keeps it the same. Which nitrogenase bases are pyrimidines? Well, we have thymine and cytosine and uracil, so we can see that is C. However, the purines are adenine and guanine. For DNA replication, bacteria was investigated. Bacteria were grown in a medium with only heavy nitrogen N15 until all the bacterial DNA was heavy. Now, these bacteria were moved from the heavy nitrogen medium and cultured in a medium with only light nitrogen N14, and this formed the first generation. The bacteria continued to reproduce, and a sample of bacteria was collected from the second generation and the DNA analyzed. Hybrid DNA contains heavy and light DNA. Which row is correct for the second generation? Percentage of heavy DNA strands. So for the second generation, basically of them being grown in N14, basically they're going to be only 50% hybrid. The first generation was 100% hybrid. The second generation is going to be 50% hybrid, meaning part of it's going to be like only containing N14, okay, and then part of it's going to be hybrid, meaning it has one N14 strand and one N15 strand. That means basically that you're going to only have, so if we have a total of like this much strands that are basically half are hybrid and half are N14, then you're only going to have 2 hybrid out of 8 total strands, right? So basically the percentage of hybrid molecules, well, we have 50% are hybrid. The percentage of heavy DNA strands, which are N15, will only be 25%, because 2 over 8 times 100 will equal to 25%, and that's why the answer is B. 24. The sequence shows the series of bases at the start of a gene, and after transcription, mRNA was translated via tRNA into a sequence of amino acids. When this part of the polypeptide was analyzed, it was found to contain the amino acids in the table. So we have 2 alanine, 3 glycine, 1 methionine, and 1 valine amino acid. So what's the sequence of amino acids in this part of the polypeptide? So here we're gonna try to and translation via tRNA when this okay, so let's go ahead and use the genes. So we can see that there's only, first off, TAC, this is a known code and it codes for methionine. It's a star, actually this is the known codon, it's a star's codon, and so first we have methionine. We can see in all options it's methionine. Interesting. Okay, so we're done with methionine. Now we have, let's see if we have any sequences that are repeated 3 times. So we have CCA, 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 so that means that this is glycine, right? So we have glycine to be this, not here, it's actually there, so we can cancel out C and D, so it's glycine or glycine, and then we have another one which is glycine, so this can also be cancelled, A, and so the answer is B. Now 25, the table shows the mode of action of 2 antibacterial drugs that can affect the synthesis of proteins. So we have antibacterial drugs, the mode of action is for rifampicin, binds to RNA polymerase. However, for streptomycin, it causes errors in translation. So if bacteria are treated with the drugs rifampicin and streptomycin, what will be the immediate effects? Transcription will stop, but the faulty proteins may continue to be synthesized. Well, that is correct, because if because it doesn't really stop, streptomycin does not stop translation. However, transcription does stop, so that is correct. If translation has started, proteins may be faulty. Well, that is correct too. And translation may be inhibited, not necessarily. No, it doesn't stop the translation. 1, 2 only, which is B. Okay, 26. The photomicrograph shows a cross-section of a leaf vein and some neighboring cells. Which structure contributes to the mass flow by the use of proton pumps? That's going to be the companion cells, which are basically part of, this phloem. And so basically the phloem over here, we can see that it is C, because we can identify A is the xylem, and then D is forming part of the epidermis. However, B, I'm not really sure what kind of cell B is. Maybe it's another cell that is part of the xylem. And then we have C, which shows the smaller cells. I would assume these are the phloem cells. So this diagram shows some tissue types in plant organs. Which row identifies the tissue types? So 1 is the phloem, because this is a root, so we can immediately identify these sections as root, stem, because we can see that the vascular bundles are arranged in a ring close to the epidermis, and then the cortex is really narrow over here. However, the cortex here is really thick. Okay, and then here this is going to be a cross section of a leaf, and we can see here that the xylem is number 4 and the phloem is number 3. So 3 is the phloem, 2 is the phloem, 1 is the phloem, and 4 is the xylem. So the answer is A. Which description of xylem vessel elements is correct? Our cells are joined to form a tube, pits at intervals, sieve plates between cells, surrounded by endodermis and roots. So, well, actually no, because we have no sieve plates, okay, in xylem vessel elements. Contains cells joined end to end, containing cytoplasm, immediately no. Contains elongated cells with end walls broken down, located in vascular bundles in the stem and leaves, that is correct. Dead elongated cells dignified with pits at intervals associated with companion cells, there is no association with companion cells, the answer is C. 29. Which properties of lignin are important for the function of xylem vessels in the stem of a tall plant such as a tree? So the properties of lignin that are important in xylem of a tree, well, basically it's inflexible, so it does not bend easily, that is correct. It's not permeable to water, really important to avoid the leakage of water. It's strong to resist collapse under pressure, very important too. It has weaker adhesion to water molecules and cellulose. Well, this is not really important, actually. We need to have 0 adhesion, so that's why the answer is 1, 2, and 3 only. Which row is correct for sucrose concentrations and water potentials in a source and a sink in a potato plant at 12 p.m.? The sucrose concentration in the source, well, obviously at the source region compared to the sink, it's going to be a higher concentration, right? And I would assume even at 12 p.m. it's going to have a higher concentration, hence the water potential is going to be lower. And so the answer, higher, lower, lower, and so higher water potential, that's going to be B. Now 31. The diagram shows one possible way in which sucrose may be loaded into a sieve tube element. Which steps require ATP? The steps that require ATP: 1. Protons move out of companion cells. So we can see this is So when protons move out of companion cells, so here this requires ATP because this is going to be active transport by pumping of hydrogen ions against the concentration gradient out of the companion cell into the cell wall. Obviously 1. Now 2 does not require energy, actually. 2 just requires the energy released from the movement of protons. So protons, when they move into the companion cells, they move down their concentration gradient. And so the energy is going to be converted, the energy of their movement, kinetic energy, will be enough in order to move the sucrose molecules with them. Right, and so basically this is indirect active transport, because the energy that is released is not needed for the movement. There's not going to be is energy released from hydrolysis of ATP is needed for the pumping of protons, not for the movement of sucrose. However, sucrose is going to be moving by using energy coming from the protons, if that makes sense. And so the answer is 1 only, which is C. Which statements about arteries are correct? Arterioles can resist high pressure, that is true. Arteries pump blood out of heart, absolutely not. Blood in the arteries has the same flow rate as veins. Well, blood in arteries does not have the same flow rate, absolutely not. If you know the graph, something like this, the flow rate is going to be the lowest, obviously, in capillaries, however it's lower in veins and arteries. And basically this is artery region, capillary region, vein region, and this is the flow rate over here. This is the flow rate. I hope you understand this. Now basically the pulse in arteries is the result of a surge in blood that causes the expansion of the artery wall. That is correct. Semilunar valves at junctions of arteries in the heart, no, that is wrong, because the semilunar valves are only present in junctions and veins. However, the only semilunar valves available in arteries are the ones between the heart and the arteries. So the answer is 1 and 4. 1 and 4, and what else? Well, with the hearts. Oh yes, that is correct. 1, 4, and 5. The answer is C. excuse me about this. Actually, this is correct because it's at junctions with the heart, not within the arteries. Basically, what happens during ventricular systole in a mammalian heart? So during ventricular systole, the atrioventricular node transmits an electrical signal to the apex of the heart. That's before ventricular systole. Ventricular systole happens when the electrical signal travels from the apex of the heart up the walls of the ventricles, such that the ventricles contract from bottom to the top, which is what happens in ventricular systole. So that's wrong. Pressure in ventricles drops below the pressure in atria. Absolutely not. Pressure in ventricles will rise way above the pressure in atria, and that's the reason that there will be shutting of the atrioventricular valves, because the pressure is going to be so high that it's going to be pushed back. Now, atrioventricular valves close and the sublunar valves open. Exactly. So it's 3 only, which is D. A shortage of protein in diet in children can lead to a disease that causes excess tissue fluid to build up in the abdomen. What explains why a buildup of excess tissue fluid can occur in this disease? A shortage of plasma. So we know basically that the excess tissue fluid is going to be because of the water potential of the blood plasma. Okay, and so what happens basically is that the water potential is going to be really high of the blood plasma. So a shortage of protein in diet causes a weak cardiac muscle. No, okay, that's not the point. A shortage of protein in tissue fluid causes a decrease in its water potential. Absolutely not. Shortage of plasma proteins increases blood water potential. Less tissue fluid returns to the blood. Exactly. To clarify, red blood cells may contain a molecule known as 2,3-bisphosphoroglycerate, 2,3-BPG. When 2,3-PPG binds to hemoglobin, a higher pressure of oxygen is needed to bring about 50% saturation of hemoglobin with oxygen. So which statements about the effect of 2,3-PPG are correct? Basically, 2,3-PPG in red blood cells causes the oxygen saturation curve to shift to the right. Well, yes, it does actually, because it's going to decrease the affinity of hemoglobin to oxygen. So I would say yes. Basically, it will reduce the Bohr effect. No, it will only reduce the affinity of hemoglobin. So basically, as I mentioned, it lowers the affinity of hemoglobin to oxygen. That is correct, and that's why it shifts to the right. That's just another way of saying this statement. Okay, so 1 and 3. And when 2,3-PPG is not present, oxyhemoglobin is less likely to unload oxygen. No, it is going to unload oxygen at high concentrations of carbon dioxide, and so I would say 1 and 3, which is B. 36. Carbon dioxide is continually produced by respiring cells. It diffuses into our blood cells and associates into hydrogen ions and hydrogen carbonate ions, and hydrogen carbonate then diffuses out of cells in gas exchange of chloride ions in the chloride shift. So what's the importance of chloride shift? It maintains a balance of, it helps to maintain a neutral pH. That's not the point. It maintains a balance of positive and negative charge between the cells and plasma. That is exactly the point. It causes the oxygen saturation curve to shift to the left. It does not have any effect on oxygen saturation curve. It prevents carbon dioxide combining with hemoglobin, allowing hemoglobin to combine with oxygen. No, it does not. So I think it's B. Okay, 37. The Potomac graph shows a section through a bronchus. Which annotated label is correct? Cartilage keeps the airways open. Well, this is not really cartilage, this is a blood vessel. Okay, D. Squamous epithelial cells are not squamous epithelial cells. Alright, elastic tissue stretches. When this is cartilage, this is not elastic tissue. And smooth muscles changes the diameter of the airway, so the answer is A. 38. Penicillin is an antibiotic used to treat infections caused by bacteria. Which events explain how bacteria become resistant to penicillin? So we have the mutation for resistance to penicillin is always caused by the presence of penicillin. That's wrong, because it's a random mutation that happens suddenly and leads to bacteria being resistant. Bacteria with the allele for resistance to penicillin pass the allele to their daughter cells. That is correct. But how do they become resistant? I suppose, okay, resistance to penicillin is a result of a mutation of bacteria. That is correct. The number of bacteria with the allele for resistance to penicillin increases. Another one, right one, so the answer is B. 39. Which statements explain why some vaccines can be taken by mouth, but tuberculosis TB vaccine has to be injected? Macrophages present antigens in vaccines to stimulate an immune response. This is a correct statement. However, this is not the reason why the vaccine has to be injected and not taken in by mouth. So this is wrong. The TB antigens necessary to produce immune response are proteins which would be digested in the stomach and small intestine. So this is a correct statement, because things like insulin, things like TB vaccine, as they mentioned here, if they're made of protein, they should not be taken in by mouth, because you know that there's trypsin and pepsin, like trypsin in the small intestine, pepsin in the stomach. These are proteases, protein digesting enzymes, which are going to digest and break down the TB antigens, and hence there will be no immune response against it. Once the antigens are present, then macrophages will be able to identify this as a non-self antigen, and they're going to carry out basically antigen presentation by phagocytosis and everything. And so this is not going to happen if the antigens are going to be broken down, right? And so this is the correct statement. No, moving on. There are no B lymphocytes and T lymphocytes in the stomach. This is a false statement. This is not the reason that it's not taken in by mouth. Like, it could be taken in by mouth, and then basically like antibodies can move in right into the stomach. The B lymphocytes and T lymphocytes do not have to be there necessarily, and if they were there, then we don't really study this as part of our syllabus, so we shouldn't just immediately assume that, oh, this is right, because we do not know. However, what we do know is that some immune antibodies, they can enter, however difficult it is, but they can enter into the stomach and into the mouth, and that's why basically, despite some people having cholera, they still manage to, the vaccine somehow 50% is 50% effective, right? And that's because some antibodies at least manage to go there, despite like the huge antigenic concealment, they still manage to reach the small intestine and the stomach, and that's why this is not a reason why it must not be taken in by mouth. Okay, so that's wrong. So the answer here is D. For stating throughout the following statements about the production of monoclonal antibodies, which statement is not correct? Non-self antigen is injected to the mammal. That is correct, actually. Plasma cells secrete a specific antibody in response to a non-self antigen. Yes, that is correct. Okay. Selected hybridoma cells divide by mitosis to form a clone. That is correct. T lymphocytes are fused with cancer cells to form hybridoma cells. They're not T lymphocytes. They are plasma cells produced by B lymphocytes. And so D is the incorrect answer. And that's it for this variant. I hope this video was useful. If you found it useful, please make sure to share it with anyone who might need it. So yeah, good luck everyone on your tests. Goodbye.